cloud. Okay.
Yeah.
Okay. So by the way, everybody's getting recorded. So you're on the record.
Are we all on it?
No.
Are we counting down?
Okay. All right. I think we're all set. Okay. My gosh.
Are we recording or should we?
Yes, we are recording. So, okay, so my pleasure to introduce everybody to Joe Gunn. So Joe has received his PhD from University of Missouri, actually in 2021, where he's worked on basically the population genetics of and genomics and conservation of endemic smallmouth bass, and has been working on hybridization. And I'm sure we'll hear more about that as well. He also has an interesting background in that he received his bachelor's degree in biology, mathematics, and minored in Spanish. So that's a very great fit for the lab, where it almost seems like everybody has to speak Spanish in my lab.
I'm rusty.
Yeah. So he's published in journals like Conservation Genetics, Ecology and Evolution, Hydrobiologia, and then currently working as a postdoc looking at Drosophila melanogaster aging, right? Or
It's coordinated evolution
coordinated evolution.
with
Right, okay.
multiomics data on, we've got phenotype data, we've got genomic data and RNA-seq data. So transcriptomics that we're looking at coordinated evolution in different diet treatments, looking at underlying haplotypes or phenotypic plasticity. So
Okay, cool. So his interests are molecular ecology, phylogeography, adaptation, fitness, hybridization, speciation, and increasingly epigenetics. So
many
he
things.
is the second candidate for our postdoctoral position in epigenetics of insecticide resistance. With that, I'll hand it over to you, and hopefully I've got this part of it under control.
Thank you, Yolanda, and thank you all so much for being here. I have had a great experience in Vermont already and traveled to several of your coffee shops, and that's one of the most important things to me and where I end up living. So I came from a conservation genetics lab. Dr. Lori Edwards group works on any and all taxa from elephants to microbes, and I happen to fit in with a project looking at the ever popular endemic sportfish, the smallmouth bass, and particularly the population genetics and their divergence and admixture between the two recognized subspecies of smallmouth bass. Go ahead.
Uh-oh, no, okay, there we go.
Oop, one back.
One back.
There we go.
Okay.
And so I often think about the question of conservation with my background. And I think that often in science, we start to ask very specialized and very interesting questions. But I always try to really think back to the first principles of why do we do what we do? Why, in my case, do we want to conserve? And I think one of the primary reasons is just the overall beauty of biology and the variation that exists. And this quote by the late poet Gary Snyder in 1990 really has resonated with me over the years as I've gone through my graduate and postdoctoral program. And he says, the extinction of species, each one a pilgrim of 4 billion years of evolution, is an irreversible loss. The ending of the lines of so many creatures with whom we've traveled this far is an occasion of profound sorrow and grief. And I think there really is just this intrinsic value in nature that's we all know exists. We all know that it's important and that we should aim to conserve biodiversity because of the beauty of variation and the beauty of the evolutionary process. But it really becomes a very complex question and an even more complex answer of why really should we do conservation? And so here are 4 examples of what can be considered keystone species in their relative ecosystems. The giant sequoia up there on the top left, the striped bass, the Atlantic blue crab, and the sea otter. And what I mean by keystone species are those species that, given their relatively small population size, have a disproportionate trophic effect on their ecosystem, such that removing those organisms might have a very large, substantial, and cascading effect on ecosystem dynamics, whether that be in terms of competition or predator-prey dynamics and the mediation of population sizes of different organisms. And so I think one of the main biological and genetic reasons that we conserve is to preserve small at-risk populations, and in particular, those populations that might be keystone species and that might have a disproportionate effect on their ecosystem. Also, the preservation in that vein of entire ecosystem stability. I think that conserving those really important species, and really just any species that is a component, an important component of its ecosystem, is so vital to maintaining ecological stability. And then there is, of course, the philosophical argument that species just have the right to exist because they have come this far with us in the evolutionary process. But I think we can't forget about the economic importance and the cultural and societal value of some species. And so I think also we need to shift our framework or to broaden our framework to include those species that are important for agriculture, to include those species that are important for ecosystem services, and for the production of services and valuable resources for our own sustainable future. And those include things in agriculture, plants in agriculture, culturally important plants, sport fish, as in my case with the smallmouth bass, pollinators and their plant hosts, and in other agricultural crops. And so I think I forgot and left this slide in there, but what I'm intending to show, go ahead and go to the next slide. Yeah, what I was intending to show here is that because of the effects of humans on the environment, some ranges of some species have been altered. So humans have had a drastic effect on the ecosystems of species by altering their native ranges. And another impact that humans have had on the environment is through the process of species introductions. So taking species from their native range and introducing them intentionally or deliberately into the native range of another species and outside of their own native range. And I love this photo. This is actually an image of aerial stocking that's happening in the mountains in Utah. Not sure what type of fish that is, but literally you're seeing a low altitude plane dumping a crap load of fish into a lake to stock, presumably for increasing the recreational fishery size. And so one potential effect that moving species from their native environment into a non-native environment is the hybridization process that can occur between closely related species. So imagine that we have this red fish who is very well adapted and well suited to its red environment. If we take those fish for some ecologically or economically important reason and we move them into this population of blue fish, which are very well adapted to their blue environment, then we might end up with genetic homogenization of that blue population that was originally blue. And then when it mixed with red, it became hybridized. And over time, if back crossing occurred between the hybrid generations and the parent generations, potentially genetic homogenization through the process of introgression. And so now we might end up with, for example, purple fish on a blue background that aren't nearly as well adapted, and there may be population viability consequences of that. And so it's these sorts of genetic consequences that we need to consider when thinking about the preservation of ecological variation and, in this case, genetic variation. Because if we lose the purple fish population, then ultimately we've gone from having two distinct populations and biodiversity to having a single population and local extinction of another. And another impact of invasive or of introducing species into another environment is simply the invasion process, or the process by which not only genetically are they hybridizing with other individuals that are closely related, but they may be outcompeting other organisms in their non-native environment. They may be predating on those organisms, and they may be altering ecosystem stability and ecosystem processes by just simply the invasion process. And this is just an example of the Asian carp, which has been widely or widely become invasive throughout the United States and much of the world.
Advance.
Yes. And we also have to think about the effect of pest infestation. And I think this is where my interests really overlap with the work that's going on in the Chem Lab. And one thing that humans also do is move agricultural crops from one place to another. And what we can see happen with that is the invasion of pest organisms that previously did not come in contact with those old agricultural crops, but when those agricultural crops moved into new geographic areas, they were able to then invade, in the case of the Colorado potato beetle, and in becoming an invasive pest on potato plants. And so now I want to really delve into my study organism and what I studied for my PhD, and that was the smallmouth bass, Micropterus dahlemi. And this is a very widely favored and sought after sport fish in the realm of angling. And the smallmouth bass has a very large endemic range that stretches all the way from the Arkansas River Basin down here in the sort of tri-state area, we call it, of Oklahoma, Arkansas, Missouri, and just a little bit of southeastern Kansas, all the way up north and east to southern portions of eastern Canada, and of course into the northeast. And I've highlighted where we are right here, which smallmouth bass are native to our lakes and rivers here in Vermont. But what I didn't know until I got into grad school, and what has only recently within the last 30 or 40 years been genetically described, are two distinct subspecies within the smallmouth bass species complex. And those are the northern smallmouth bass, which stretches all the way from the Arkansas River Basin, but does not inhabit the actual Arkansas River Basin. It's just adjacent and parapatric, all the way up north and east to the entire portion of its native range. And then the much smaller and geographically restricted native Neosho smallmouth bass, which only inhabits or is only known or recognized to inhabit streams of the Arkansas River Basin in the central interior highlands, which is what CIH there stands for. And overall, smallmouth bass are ecologically important for ecological stability. They are top apex predators in their environment, which means that they control top-down dynamics. And they are also hosts for several invertebrate organisms, particularly different species of mussels. And it's actually the specific species of mussels, Glaucidia, is a good example of one that inhabits the gills of smallmouth bass as part of their essential part of their life stage. So without smallmouth bass, we wouldn't have the mussels and vice versa potentially. And they're also economically very, very important. This is just a cartoon image of some, but not all, of the Bass Pro Shop organizations that have been constructed over the last 10 years. And Bass Pro Shops, of course, is a multi-billion-dollar industry that is focused on all things hunting and fishing. And so millions and billions of dollars every year throughout the world are spent on equipment for fishing, on traveling for fishing, and for all of the above for hunting and other outdoor sports. And so smallmouth bass, as a native endemic fish, really do support and fuel a multi-billion-dollar recreational industry in the United States alone. And so I come to this question of how do I use genetics and genomics, and how can we, the scientific world, use genetics and genomics to understand the ecology and evolution of species and for the conservation of biodiversity, and in particular for the conservation of economically important species. And I take a three-pronged approach, which I think can be applied to most economically and ecologically important species. And the first step, I think, in the process is to identify ecological patterns in the environment. And we can do this by looking at genetic and morphological patterns between different populations within a metapopulation or just within a species. We can see how individuals are genetically or morphologically related and how those mechanisms might contribute to their role in the respective ecosystems. And then after inferring those genetic and morphological patterns, we can start to gain a much clearer picture and understanding of the evolutionary processes that have driven or contributed to those patterns. And then once we understand the evolutionary processes that have driven those genetic patterns, then we can start to figure out what are the actual fitness consequences of those evolutionary processes and the current ecological patterns that we see. And then how can we use those to understand the long-term viability of populations, the long-term sustainability, and what we can do to help increase that sustainability. And so in the context of the smallmouth bass, for really what became or culminated in my first chapter of my PhD, I was interested in understanding what is the genetic divergence between the smallmouth bass subspecies that we currently recognize. Because at the point of their description, they were really only considered different subspecies based on subtle morphological traits, where if you put one up to the other, it looked like they may be different in size, they may be different slightly in coloration, they may be slightly different in the presence or absence of certain numbers of meristic traits. It was all very subtle and very, the waters were muddied a little bit. So I also wanted to understand what geographic distribution of diversity looks like in the smallmouth bass. If there are distinct populations, if these subspecies are distinct, do they come out as geographically being distinct? And then are there distinct populations within the geographic ranges of the Neosho and the northern smallmouth bass subspecies? I also wanted to look for, if they existed, patterns of intermixing between populations. One thing that really happens with smallmouth bass, like many economically important species, is their movement, of course. And so I wanted to see if we could find signatures of hybridization that could possibly be the result of anthropogenic stocking or the movement of individuals from a hatchery population into a native population. And I also wanted to look at morphological differences to really get an idea of the subspecific status of these subspecies. And so previous work has shown that there is some genetic differentiation between the northern and Neosho smallmouth bass. What you're seeing here on the left is a principal component analysis of a very small number of allozyme loci back in 1998 by Stark and Kelly. And what they showed was that based on those allozyme loci, there was clustering of the northern smallmouth bass here in purple triangles, and they're shown geographically here in the adjacent portion of the northern native range to the Neosho native range. Here they showed clustering of the Neosho smallmouth bass that were genetically differentiated from the northern, and they're shown in these sample sites in green in the Arkansas River Basin as expected. And they also found genetically divergent individuals within two separate basins, shown by squares here and upside down triangles, within what are known as the Ouachita Mountains. And so there's potentially even 3 distinct lineages that had originally been described. But the morphological descriptions were a little bit less clear than this initial genetic evidence. Originally, the smallmouth bass were considered to be slightly different in, go ahead, in total length. And so it was essentially thought that the northern smallmouth bass tended to be a little bit longer than the Neosho smallmouth bass on average, but not really tested. It was more based on some visual and anecdotal evidence. And the original descriptions, by the way, were done by Hubbs and Bailey in 1940. They also varied a little bit in vertical bar patterning along the lateral side of the fish. They also varied potentially in the number of soft dorsal fin rays, the individual number of rays in the soft dorsal fin complex, which is behind the sharp fin concept or complex. And so ultimately, to understand better the genetic divergence, genetic population structure, and morphological divergence between the smallmouth bass subspecies, I wanted to use more nuclear markers than what had been used before, and to get a better holistic picture of nuclear population structure and morphometrics to both holistically look at morphological differentiation overall, but then also differences corrected for the total length of fish of individual morphometric traits. And so just as a primer of microsatellite loci, and what they are neutral sites for the most part in the genome, meaning that they're generally not seen by natural selection, and so we can use them to understand geographic patterning of populations. And different alleles are just different numbers of short tandem repeat units within the genome. And so what I'm showing here is the study area where I did my study, in addition to the entire geographic range of the Neosho and northern smallmouth bass for reference. And I collected sites or collected samples from approximately 43 sites throughout the native range, some of which have been excluded here and clumped into larger populations for the sake of visualization. Individual circles here are large because I'm going to fill them with pie charts that show the geographic clustering from microsatellite loci. But ultimately, we collected 838 smallmouth bass individuals from across the Neosho native range, and then in the adjacent portions of the northern native range, accounting for individuals in two different drainage systems. This is the White River drainage, which flows southward, southeast into the Mississippi River, but much lower than the Missouri River tributaries, which flow northward into the Missouri River and eventually into the Mississippi allopatric populations within the north. But then also we included this lake population, Skiatook Lake in Oklahoma, which we knew previously to have been stocked with a northern strain or a presumably northern strain of smallmouth from the Cumberland drainage system in Tennessee. And so we found that several sites within our study were what we call genetically pure, where most individuals within that site on average assigned mostly to a single genetic cluster. Anywhere where you see green in these pie charts, that represents Neosho smallmouth bass. Anywhere where you see purple is where we inferred northern smallmouth bass. We also used spotted bass as an outgroup. So we did collect 30 spotted bass that we used for phylogenetic comparisons later on in our study. And what we found was that several populations, 6 to be specific, in the Neosho range were mostly genetically pure, where most individuals assigned to a single genetic cluster that were differentiated from the northern smallmouth bass at the optimal number of populations inferred in our population structure analyses. But you can see that in each of these sites, there are traces of spotted bass alleles that might indicate some interspecies intermixing or admixture that we had not really predicted that we would see. And we also included a hatchery in Wisconsin that came back to totally assign with the northern smallmouth bass preferred individuals as well. And then we found many populations only within the Neosho range that came back as heterogeneous genetically and showed variable amounts and strengths of admixture with the purple or the northern range individuals. And I say heterogeneous because we did not see a consistent pattern where we saw a certain amount of northern smallmouth bass ancestry. They were variable, some having a little bit less than others, but we did see this signature of admixture in only some of our stream populations. Overall, we did also find that the Neosho smallmouth bass were less diverse in heterozygosity and private allelic richness than northern smallmouth bass, indicating that this probably falls in line with the small population size of the Neosho smallmouth bass and might be indicative that there is lower genetic diversity because of that small size and that there may be an impact from admixture as a result of it. And for morphological analyses, we collected multiple morphometric traits, including the total length of the fish, which we would ultimately compare all of our morphometrics back to. We took the standard length of the fish, which is similar to the total length. The total length, by the way, is from the tip of the lower mandible of the fish all the way to the back of the caudal fin. Standard length comes back to the hyperal plate, which is this sort of cartilaginous structure in the back portion of the fin just before the caudal fin begins. We took head length, which was the length from the lower mandible to the end of the gill plate. We took orbital diameter, which was just the diameter of the eyeball. And we took the body depth. So at the deepest portion of the fish, we measured how deep and how broad its body was. We also took one morphometric trait, and that was soft dorsal fin raise, to really test whether or not there was a diagnostic difference between the number of meristic traits in northern and Neosho smallmouth bass. And what I did not highlight earlier was that the Neosho are thought to only have 13 meristic rays, whereas the northern are thought to have 14. So it's really only a single meristic trait or a single number difference that we really wanted to see if it was diagnostic between the different subspecies. And what we found morphologically was that the one meristic trait that differed between northern and Neosho smallmouth bass, accounting for the total length of the fish, was head length, where Neosho individuals, for their body length, tended to have larger heads. And this again was the length between the edge of the lower mandible to the back of the gill plate. But unlike this significant difference in this morphological or morphometric trait, we did not find that there was a diagnostic difference in the number of meristic soft dorsal fin rays between the Neosho and northern smallmouth bass using a logistic regression analysis, where we found that approximately a relatively large proportion of individuals that were Neosho did have 13 rays, but we certainly could not classify individuals well or accurately based on the number of rays that they had. And we used proportion of Neosho ancestry as our predictor variable there to determine whether or not those that were more genetically pure Neosho might be more likely to have 13 versus 14 rays. So some higher likelihood, but not diagnostic by any means. And so from all of that information, from these differential patterns of admixture and differential patterns of genetic differentiation that we saw within the Neosho and the northern smallmouth bass, and the morphological differences, we wanted to start to understand the evolutionary processes at work driving these patterns. So ultimately, we wanted to look at evolutionary diversification. How, to what extent are they genomically different, the Neosho and the northern? We wanted to look at ecological drivers of divergence and local adaptation to really look for evidence of selection happening at the local stream level if we could find it. Natural biogeographic mechanisms of evolution. Here, we wanted to use demographic analysis to see if we could figure out at what relative times were these different streams admixed that we found to be admixed with the genetic data. And then we wanted to be able to infer potential anthropogenic mechanisms in comparison to those biogeographic mechanisms to look at, are we seeing very recent admixture versus potentially historic admixture that may be due to other geological or ecological mechanisms. And so just to give you an image of where I did most of my field work and to highlight the potential for local adaptation, this is an image of the Ouachita Highlands here down in central, actually this is the Boston Mountains rather. This is in the northern and central portion of Arkansas where the Neosho range really comes down to its southern edge. And it's very hilly terrain, it's very karst terrain, and it's relatively warmer down in the Boston Mountain range than it is further north in the Neosho range. There's a pretty heavy temperature gradient there. Very variable precipitation amounts in all sorts of different ways that fish could be experiencing differential selection pressures. And I just wanted to highlight the sort of beauty of the streams where I did most of my field work, which was indeed just fishing for the most part. And that over there on the right-hand side is my first undergraduate assistant, Caitlin Solkowski, who was a much better angler than me, caught all of our big fish. And this was actually a fish that she collected by electrofishing. And electrofishing is a method for collecting a higher volume of fish, where you literally, we did boat electrofishing where you have prongs, electrically charged prongs that dangle into the water off your boat, and you can zap the water and they go bloop, 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 and then you can collect it. It doesn't kill them, so but research on the effects of electrofishing on fish is ongoing. So, but we were able to sample higher volumes of fish in that way. So we had two main hypotheses for admixture patterns that we saw using the nuclear genetic microsatellite data. One of our hypotheses was recent anthropogenic introductions, where I just have as a cartoon that the fish are being stocked from Skiatook Lake, which we know is a stock population. Presumably they could be coming from any hatchery that exists, any national or state hatchery. But we could see anthropogenic introduction signatures if we were able to infer very recent admixture timing. Our second hypothesis was historic timing of admixture, where we might infer based on demographics that if we have older admixture events, then those admixture events may actually have been caused by things like stream capture, where one stream, simply due to erosion and other geologic uplift mechanisms, flows and starts to flow into another one or capture another river. And so this was important that we make this distinction between anthropogenic stocking and natural biogeographic processes, because if natural biogeographic processes have been responsible for this admixture, that has much different implications for the evolutionary process than the types of evolutionary mechanisms that we could potentially intervene in if necessary to preserve biodiversity. And so to address these questions, we used, go ahead, DD-RAD-seq methods, and did not at the time have a closely related reference genome. And so all of our DD-RAD-seq was done de novo to look for patterns of SNPs across the entire genome. We wanted to look for outlier loci, specifically using FST as our metric of interest, outlier FST values, to look for signatures of selection and local adaptation in different sites. And we wanted to use the site frequency spectra, specifically the joint site frequency spectra between pairs of populations to look at demographic history and admixture timing. And so just as a quick primer of DD-RAD-seq, it basically is a method by which you use 2 restriction enzymes as opposed to just 1 in your standard RAD-seq methods to digest fragments of DNA randomly throughout the genome at restriction sites that are of particular length. And so it is able to focus in on particular fragments of DNA that we can identify single nucleotide polymorphisms between different individuals. Ultimately, we found 50,828 SNPs that were variable, of course, between individuals across all of our 88 smallmouth bass that we used within the Neosho and Northern Range. And again, I've shown circles here to show sites where we collected from and did our genomic sequencing with. And I will fill them with our phylogeographic inference here. And so what we found was we did actually an initial screen of all of our individuals to see individuals that were putatively admixed in different sites, in which we considered individuals that had greater than 5% ancestry for one subspecies or the other to be admixed. We excluded those from this analysis, and we looked at just those that were mostly genetically pure to look at phylogeographic differentiation among the different populations. And we were very interested to see that not only did the northern and Neosho smallmouth bass form reciprocally monophyletic groups at all 58 or 50,000 SNPs, but also within the northern range, there were 2 reciprocally monophyletic groups in this dark pink drainage here and this dark blue area here that included the Missouri River tributaries, but also this stocked population in Skiatook Lake. And so we weren't terribly surprised to see that geographic distinction there, given that they're allopatric populations leading to potential divergence at different SNPs. But we were very interested to see that based on our structure analyses, there was this split between the dark green populations in the Boston Mountains and these light blue populations in the middle and upper Arkansas River Basin. They did not come out to be reciprocally monophyletic groups, but based on structure analyses, they were potentially distinct. And so we wanted to look more into these potential, processes that might be leading to local adaptation. And so we did even further population discovery using a program called FineRAD structure, which actually uses specific haplotype differences by concatenating different SNPs within individuals to look at single nucleotide differences. And we found even finer scale population structure within the northern range within the White River drainage, the Missouri River drainage, and separately in Skiatook Lake. We also found 7 populations that were significantly admixed based on admixture mapping analysis. And this is when we added those potentially admixed individuals back into the analysis. And considering all of those populations, we wanted to look at signatures of selection. So we looked at outlier FST analysis. So we did this on 2 hierarchical levels, one being the whole subspecies level, where we considered SNPs for all northern and Neosho smallmouth bass. And we found 156 SNPs that significantly differentiated individuals from the northern smallmouth bass range from a much variable spread of individuals within the Neosho range, but 156 SNPs that were significantly diverging those on the first principal component. We also did this at just the Neosho level to look for potential signatures of selection within the Boston Mountain region. And what we found was that not only is there strong differentiation between the upper and middle Arkansas populations from all of the Boston Mountain populations, but also that there was strong differentiation on the second principal component between the Lee Creek, which is this river here, Mulberry Creek populations, separately from the Illinois Bayou and Big Piney Creek, Arkansas rivers. So we were seeing this multi-hierarchical signature of population structure at these very strong high FST outliers, showing potentially divergent local adaptation within these streams. And just looking at neutral SNPs, just for fun, we wanted to see if there was a large signature of genetic drift. And we did find that most of our populations were able to be completely distinguished and completely clustered from one another, and they're colored just as they are there on the principal component analysis. We found 35,038 SNPs that were putatively neutral that showed these differential patterns of genetic drift. And so we also wanted to use site frequency spectra from pairwise populations from our inferred source populations for admixture to look at relative timing of divergence and admixture timing within two different populations. So what I'm showing here is a cartoon schematic of what the results from our analysis looked like. And so you can consider that we start with one ancestral population that splits into two different populations. And the null model is that we have a split or a divergence between two populations with no admixture between them. And so from this analysis, we can estimate the time since that divergence occurred using site frequency spectra. We can also infer the non-null model of admixture or migration in one direction or the other between different populations. It either goes both ways or just asymmetrically one way or the other. And then even further, we can break this down by timing. So we can estimate the timing of these admixture events as either ancient or as relatively recent. And then ultimately, we can see what the demographic patterns show us about admixture dynamics. And what we found is that those populations that were light pink before, which are the Spavinaw Creek here, Big Sugar Creek, and Buffalo Creek, were most likely admixed from alleles or individuals from this White River drainage signature, this dark pink signature. And we also found that that admixture occurred widely or broadly between two different distinct epochs of time. And what that analysis actually shows is that there were differential amounts of admixture between two different sets of time. So that it's inferring differential amounts of admixture across that divergence time. But all of that admixture, for the most part, was asymmetric and coming from the dark pink White River alleles into the Neosho system. For just the Elk River, we found an even more interesting pattern where we found continuous admixture over time, again, that was asymmetric coming from the White River drainage into the Elk River. And then interestingly, and as we actually would have expected from within the Illinois River population, we found that alleles were primarily coming from the Skiatook Lake population or from the same strain of hatchery raised fish. And we found that that admixture was very recent, which falls in line with our hypothesis of recent introductions that we actually knew in this river there had been a previous introduction of smallmouth bass in the early 1990s. And then finally, in the Illinois Bayou and Big Piney Creek river systems in the Boston Mountains, we found that admixture was again very recent and asymmetric from the White River drainage into the Boston Mountain range. And so ultimately and holistically from that, we found that there were very differential timings of admixture history, which indicates that in some cases, we may be seeing a very recent anthropogenic stocking effect. And in other cases, we may be seeing a more natural historic effect. How are we doing on time?
This is just another seminar right at 1:10. so,
Gotcha.
Yeah.
I'll go pretty quick. Okay, and so then we wanted to look at the fitness consequences of these evolutionary processes. And specifically, we wanted to look at the effect of admixture on fitness traits, the geographic extent of non-native ancestry within the Neosho native range, and we wanted to look at the correlation between heterozygosity of individuals and their overall fitness in the form of body condition to assess what the effect was of admixture on fitness. And so we know that smallmouth bass are widely introduced within and outside their native range. And that's why this idea of assessing the effect of admixture is important. We want to know, is the introduction of smallmouth bass and potentially subsequent introgression and hybridization with other closely related congeners creating increased fitness, decreased fitness, or none of the above, and what are the implications for conservation? And so introduced smallmouth bass can have lots of different potential negative effects, from just having invasive smallmouth bass that outcompete other native fishes, that prey on other native fishes, but also they can introgressively hybridize with closely related congeners. And this can lead to genetic swamping. And that is the replacement of one lineage with another entirely. So the loss of biodiversity from two different subspecies down to one. Or alternatively, it may have the opposite effect. And it may have a hybrid vigor effect on the individuals, creating more fit or hybrids that are better able to survive and reproduce in their environment. And this may ultimately then lead to genetic swamping. Or we may see outbreeding depression, the opposite of that, in which fitness is reduced in hybrids, which ultimately results in the loss or local extinction of those populations that are hybridized. And we know from previous evidence that hybridization and introgression does have a negative effect on similar or closely related species. This is the largemouth bass, which in a study in 2005, populations of parent smallmouth bass and F1 and F2 hybrids were inoculated with largemouth bass virus. And these researchers, Goldberg et al., wanted to see how survival was affected in these different hybrid groups relative to parent groups. And what they found was that F2 generation fish, those that had been introgressed, were much less likely to survive over generations than other fish in the parent or F1 generations. And so to look at these patterns, we wanted to assess the microsatellite differentiation between individuals, again, to look at genetic ancestry and to correlate admixture with fitness traits. And our fitness traits of interest were individual growth of fish, so how fish are growing over time. And we wanted to correlate heterozygosity and body condition. And so we collected fish from Big Sugar Creek and the Elk River, which were both known to be admixed with northern smallmouth bass from our previous analyses. And we went from field sampling, where I went out and caught a bunch of fish, and we extracted the otolith structure from the inner ear, which is essentially a calcareous structure that has basically tree ring-like things that you can count to estimate the age precisely of fish. And then we did DNA analysis to assess the ancestry of individual fish and then correlate the ancestry with growth. And so we found patterns of genetic admixture throughout the Big Sugar Creek and Big Sugar Creek and Elk River systems. And we divided individuals into an admixed group, pure Neosho group, and pure northern smallmouth bass group for a later analyses. And just to show you how otolith age is estimated, basically you just count the rings down from concentric circles, starting with 0 here in the middle and radiating outward. And this individual is approximately 7 years old based on multiple independent observers doing the counts. And so we wanted to look at age and how it correlates with total length of the fish. And so we collected fish of many different lengths, and we estimated their age in order to get an estimated growth curve for a population of fish. And this is done by von Bertalanffy growth modeling. And so this is a known and well-established model for how fish typically grow in different populations. And so we wanted to look at these growth patterns between admixed, pure Neosho, and pure northern fish. And what we saw was that, and I think in the image there should be little error bars that kind of come up here too, but I think they got washed out in the image here. But what you can see is that there are, and these confidence intervals are very big here. There are actually no major differences between northern Neosho and admixed fish in Big Sugar Creek or in Elk River overall, between just the Neosho and the northern. But then what you can't really see here is that in the Elk River, the purple individuals, those that are admixed, actually had a significantly lower overall maximum theoretical total length than both Neosho and northern. The confidence intervals come to about here and here there. So the average total length of the other two groups falls outside of the admixed group. And we also wanted to look at heterozygosity and fitness correlation. And so we did this using the Fulton index of body condition, where we took mass divided by the total length volumized of the fish. And that gives us just an estimation of really how bulky or how robust that fish is and how healthy it is. So we have skinny fish and fat fish. And then we can look at multi-locus heterozygosity across microsatellite markers and correlate that with that fitness measure of the Fulton index. And what we expected is that if we saw no correlation between heterozygosity and fitness, then we would see no effective admixture on the fitness of the individuals. If we saw a positive correlation between heterozygosity and fitness, we would call that heterosis or hybrid vigor, so that individuals that are more heterozygous or have more multi-parent ancestry are better suited to the environment. Or if we saw the opposite effect, we would consider it outbreeding depression, in which the increase of heterozygosity or mixed ancestry might lead to decreased hybrid fitness. And what we ultimately saw was that there was a significant correlation between multi-locus heterozygosity and body condition of our fish. And the R-squared value for this wasn't great, but the p-value was 0.03. And so this really is not a definitive confirmation of outbreeding depression, but it does give us evidence that will serve as preliminary work to continue this work into the future and to really get a fine assessment, hopefully doing genomics on interspecific heterozygosity and fitness to really assess what's going on in these rivers to see if admixture is having a hybrid vigor or a hybrid breakdown effect. And so ultimately, wrapping up, that is how we use genetic and morphological patterns, ecological and evolutionary processes, and fitness consequences to address the conservation of smallmouth bass. And so why should we focus on conserving the Neosho smallmouth bass? They're evolutionarily distinct, which we found with reciprocal monophyly in our phylogenetic tree, potentially locally adapted, at risk of admixture, which potentially is leading to lower hybrid fitness. And they're also a highly economically important sport fish, which really fuel a multi-billion-dollar industry in North America and the United States alone. And future research in this, and why I've been interested in epigenetics recently, is we also know that in the tri-state mining area where Arkansas meets Oklahoma and Missouri, there was a historic mining operation primarily for zinc and lead, which has since ceased since the 1950s. But there are known effects in some rivers within the Neosho range that these trace zinc and lead toxicants in the rivers have effects on reproductive fitness and reproductive development and other physiological traits that are similar. But what hasn't been studied is the potential epigenetic effects or epigenomic effects from these toxicants on these fish and how that might be leading to or reducing local adaptation. And so what I would be interested in knowing is if varied toxicant exposure in different streams is leading to differential epigenetic effects, and if we can use that in our understanding and movement forward in conservation. And so ultimately, I wanted to bring this back to show how it connects with the CPB, the Colorado potato beetle, because you might be wondering, how do we go from conservation of an endemic sport fish to being interested in the evolution of insecticide resistance, or specifically epigenetic resistance in beetles? Well, I think one of the biggest similarities here is that we have a very economically important problem, and that's that agriculture is of the utmost importance for food sustainability for humans. And this pest, the Colorado potato beetle, for example, is a potentially billion-dollar problem costing billions of dollars in agricultural insecticides and damage costs, which is similar to the billion-dollar industry of sport fish. And so ultimately, I'm really interested in this context of understanding what is going on with the genetic bottlenecking of Colorado potato beetles. We actually know that the European invasive Colorado potato beetle is all derived from a single mitochondrial haplotype. And this just really is emblematic of the genetic bottleneck effect that exists when applying insecticides to populations like this. So I'm interested in understanding what is it about epigenetic effects, or in specifically the interaction of hormetic effects and potentially genetic rescue effects in these bottleneck scenarios that is causing these beetles to be so successful and to evolve resistance to pesticides. And further, I want to put this into context of my population genetics and conservation genetics background. And I really want to understand, are there epigenomic patterns that are repeatable and parallel between different populations in different parts of the native range and introduced range of the Colorado potato beetle? Can we see potential local adaptation effects or similar resistance to insecticides across different populations? And do we see those because of changes in methylation patterns, transposable element mobility, transcript diversity, or any other epigenetic related mechanisms? So I have many people to thank. That I think I did go over time, and I apologize for that. I have 3 slides of acknowledgements that you can run through here that include my AgriLab family and all of the folks that I work with at the zoo, all of my friends that contributed to helping me through all of this work, and all of my collaborators, et cetera, who helped me do the fishing and the DNA work and all of the above. And with that, I don't think we have any time for questions.
No, we
But
we
sure.
do. I mean, I think so. So some of us might have to run out, right? So
Paul is a collaborator.
so yes.
That was awesome. Excuse me running out. If you take online questions first, we can't start the next Zoom until this one is.
Oh, I'm
So take
sorry.
The online ones, so that's, sorry, that's why.
Oh, I thought it was a separate one.
It's not a separate.
Oh, okay.
the
All
way
right.
our subscription works, it's
Okay.
not a separate. I do have a question for you. Sure. At dinner, I'm on the IUCN, it's a background. I'm on the IUCN.
Okay. Are there any
Conservation
online
Genetics Committee.
questions?
We are writing a new IUCN position statement on hybrids because.
Any
Plants,
questions for
plants,
online?
Yep, hybridization is ubiquitous.
Yep.
But it clearly happens. with
All right,
fish.
I
It's
think I
not
might-
all anthropogenic. It could happen.
I can see the chat
Green,
here.
capture. How do you think, so this is the third one, we should be advocating for conservation? Many of them are very broad. Are framed as all hybrids are bad. Like these are bad.
Yeah, that's a really good question. And I think that the first step is to identify what mechanisms are driving that hybridization. Because I think given the ubiquity of hybridization and its importance in the evolutionary process, I think that it has contributed as much to biodiversity in many cases as natural selection and genetic drift throughout historical time. And so by identifying what that mechanism is first, then I think we can ultimately make the decision of whether or not it is lucrative or viable to intervene in established management and conservation protocols for mediation of those hybrid populations. Because I think not only are the mechanisms causing hybridization complex, but also the effects are super complex. And if we may see an increase in fitness of populations due to hybridization or rescue, or we may see the opposite. We may see outbreeding depression, or we may see no effect at all. And then those have different environmentally dependent contexts, which can ultimately lead to genetic swamping in some cases, in the case of heterosis or genetic swamping. or they might, lead to the formation of a hybrid species and a new species entirely. So I think it's a very complicated question, I think. And it will require lots and lots of more, thought, I think, too.
It went white
Right.
Okay, for those of you that are online, I'm going to,
They are mammals.
sorry, for those of
Thank
you
you.
that are online, I'm gonna close the Zoom meeting now so that the people that are using the same Zoom account can start the next one. So I'm gonna thank you for coming. And feel free to email me if you would like to talk more or chat with the